Good afternoon, everyone. I want to thank the organizers for the invitation to present here this afternoon. The first post-lunch presentation. I'll do my best. Sentien Biotechnologies, we are a venture-backed clinical stage cell therapy-based company out of Lexington, Mass. And we say delivering on the promise of cell therapy. I think there'd be a lot of people here that are all saying we're delivering on the promise of cell or gene therapy. Where we like to emphasize, advancing the slide maybe. That was a dramatic pause on what we emphasize. I don't know if you guys can I get you guys to help me out? Yeah, thanks. There we are. Mesenchymal stromal cells. This audience, I left you hanging there for a bit. What we emphasize is on the delivery, and it's a little bit of a play on word. We're using mesenchymal stromal cells, which this audience knows has been tested in numerous models preclinically. They've been tested clinically in a number of indications, a sample of which is shown in the word cloud on the left, but collectively have not had a lot to show for it. I think there's a lot of unrealized promise out there in the MSC space. The potential of MSCs is really based around, as many of you know, the multitude of secreted factors and pathways that are being modulated by this really interesting cell type. But we think that dosing limitations have really hampered the clinical translation of these cells. And one of the arguments is that the field has been working on is how do you get more exposure of these MSC-secreted factors to the patient while maintaining the viability of the cells. So how do you get really the PK profile that you might look for? And as an IV infusion, there's data out there suggesting that the half-life is very short when injected. So you get a very a short spike of therapeutic factors being released, but you don't get a very durable response. And we believe, as do others, that this has been one of the reasons that have hampered MSC translation into a clinic. So the way we try and solve this problem is using a combination product where we're taking a hollow fiber device and we seed the MSCs in the extracapillary space of this device and allow patient blood to flow through the intracapillary space with the MSCs in the extracapillary space. So we immobilize these cells on these hollow fibers akin to an MSC being adhered to a blood vessel, and that allows for bidirectional communication between inflammatory mediators in the patient's blood with the MSCs themselves. And so what you get is a continuous perfusion of the MSCs in the blood together, and you get a different kind of PK profile than you might get as an IV infusion. So we can therefore control, obviously, the number of cells that are being administered. We also can control and observe over time what these cells are doing. So by putting these cells in the extracapillary space, we can actually sample from the extracapillary compartment and characterize, if you will, the MSC environment even during treatment. So once you inject cells via IV, obviously it's very hard to track what happens, how they're behaving, what they're producing. With this system and this architecture, we can be sampling that. In fact, we are sampling that even during blood perfusion. So it gives us a way to look at MSC activity in situ, if you will, and then correlate that with biological activity that's observed in the patient population. So we've tested this product in these hollow fiber bioreactors, and we do have viability readings that go on for days where the MSCs continue to produce factors and, importantly for cell therapy, dynamically respond to different changes in the environment from the patient's blood. So a little bit of data to emphasize that particular point. In a large animal model that we performed, we were measuring a certain MSC-secreted factor, and it was a xenogenic model, so we could measure the human levels of MSCs and characterize that in a dose-dependent way in this animal model. And then we were able to correlate that into a pharmacologic response. So I'm trained as a small molecule guy, so I say PKPD, and that means something to me that traditionally is different than cell therapy. But I think what we're trying to do is begin to create that PKPD correlation with a cell therapy, even during treatment. So we're going to be collecting quite a bit of data in our ongoing clinical trial to create these correlations and understand over time the pharmacologic relationship between drug product and patient. So in this example, we were just showing that even in a large animal model with human MSCs, there were 2 cell populations that we evaluated. One was a monocyte population, one was a T cell population that had changes in response to the MSC therapy. So a little bit more preclinical data before I talk about our ongoing trial. This was a large animal model in ischemia-reperfusion-induced acute kidney injury. That is our first indication clinically. We did this study for a number of reasons, obviously to evaluate efficacy, but also in the scale-up. What I didn't describe earlier is the design of the architecture. This cellular device is integrated into commonly used blood circuits. So if you think about acute kidney injury as one indication, the standard of care is continuous dialysis in an ICU setting, and our product is then used and integrated as an add-on to the dialysis circuit, allowing for that continuous perfusion to take place along with standard of care in the clinic. So this model was replicating or mimicking one etiology of AKI, ischemia-reperfusion-induced AKI, and what we had with no treatment, meaning it was fluid resuscitation only, there was 50% survival over 7 days. With dialysis and an acellular sham control, we had 70% survival. And with SBI 101, our lead product, we had 91% survival, again, over 7 days. What was interesting about this model is trying to replicate the clinical paradigm. We induced the ischemia-reperfusion injury and then waited a full 24 hours to mimic what you might see in a clinical setting. At baseline, these animals had a 4-fold rise in creatinine, which is very significant injury. We then administered a dose for 12 hours of continuous perfusion and then took the product away and followed survival out to 7 days. And not shown in the previous data was that as we administer these MSC-secreted factors and the blood gets conditioned with our product, the responses that take place in the immune system remain durable even after a product's been removed. So you're reconditioning or retraining the immune system, and the changes that are taking place in that immune system persist even after product's been removed. And that's somewhat highlighted here in this diagram. So we are actively enrolling in a phase 1/2 clinical study. We're looking at 24 patients in a double-blind randomized study. We're still in the low-dose cohort at this point. We do expect to have results to be shared certainly by this time next year. The primary goal obviously is safety, looking at renal efficacy endpoints, not powered for that necessarily, but just taking a look at those parameters since this is our patient population. But I think the most important component for us moving forward is understanding the pharmacology. It's this PKPD relationship I was describing earlier, finding those correlations between MSC biology and AKI pathophysiology, making those connections that we think can help us then better design another study going forward in a phase 2B. So the pharmacology is really the primary focus of this. You might call it a dosing study in that regard. Once we have more data here, we would also like to initiate a study in the liver. MSC biology is not kidney-specific per se, so we will be looking at other organ failure indications as well, with liver being, we believe, next on the docket for us. So this will be more likely an open-label study based on the fact that liver patients don't get dialysis as standard of care, but we think the underlying mechanisms of addressing acute systemic inflammatory conditions will take us similar pharmacology for AKI as it is for liver. And then going beyond that, the vision really is to think about transforming extracorporeal devices or blood circuits that are commonly used in a critical care setting for acute systemic inflammatory disorders like AKI, like acute liver failure, but also using other modalities where blood circuits are used as well. Plasmapheresis, photodynamic therapy, ECMO is something that we think is readily applicable for us looking at lung injury as well. So once we have initial clinical data, we hope that will be our launching point into thinking about other critical care applications. From an architecture perspective, the plumbing into these different blood circuits is really very trivial. The bigger challenge, obviously, is understanding the pharmacology, which we hope to be making significant progress on. So from a company perspective, going back to, again, delivering on the promise of cell therapy, we believe that if we can show in this way how MSCs might be actively and persistently delivering those secreted factors, we think that can take us, as I mentioned, from AKI into the critical care space, but then again, understanding other types of biology where you might veer off into the chronic kidney disease space. It's not lost on us the opportunity to disrupt the outpatient dialysis market and what that could mean. It is a different biology for sure around chronic kidney disease, but the potential's there, and there is some data on MSCs and CKD as well. But then looking at other types of autoimmune disease, again, further down the road. My investors would demand that I say we are focused on AKI, which we are, but we think this is a good launching point for us to go forward. So I was, I think, advised to leave time for questions, so we have a few minutes left if there are any questions in the audience.
I'll ask you one. Does the mechanism of AKI matter? Is that part of your inclusion criteria?
So if we were trying to answer every question in our phase 1/2, we would have gone just to one etiology of AKI. We're looking at multiple etiologies. There may be differences with MSCs for septic AKI versus ischemic AKI. So we're not looking to answer all those questions now. We're looking to think about our phase 2B and how we design that best. So we'll look at septic patients, evaluate that data, look at ischemic patients, evaluate that data, and maybe in our phase 2B we would want to limit it down.
Thank you.
Yep. Okay.